Welcome back to my channel. In this video, I will be discussing how to present bacterial growth data graphically. We all know the importance of making data look attractive and presentable. And when it comes to this, diagram, graph, charts are often our top choice. Graphs can convey the same amount of information as tables, but in a smart and easier way. Viewers will understand it quickly. So it's crucial to learn how to graphically represent bacterial growth data and how to determine generation time from those graphs. In my previous video, I covered bacterial growth kinetic parameters such as generation time, specific growth rate constant, along with the mathematical calculations involved. These parameters allow us to measure how quickly bacteria are multiplying. In this video, we will explore how bacterial growth can be expressed graphically and how to calculate those kinetic parameters from graphs. Suppose initially only 640 bacterial cells were inoculated into artificial growth medium and allowed it to grow under controlled laboratory conditions. What will happen? The bacteria will divide exponentially and the number of bacteria will increase in this way, and the exponential growth is characterized by the number of cells doubling within a constant time interval. In this way, when the cell number is plotted on arithmetic coordinates as a function of time, you will obtain a curve like this with a continuous increasing slope. During exponential growth, the increase in cell number is initially rather slow, like from 10 to 20 and then 40 to 80. And here the scale is 5000, and because of that, after 7 hours, you don't observe any visible changes and the line looks flat. When we magnify this portion and change the scale from 5000 to 20, now you can see the difference. The number is increasing. This is one of the major drawbacks of this curve. But later the bacterial number increased at an ever faster rate, like from 3,27,000 to 6,55,000 cells, and the difference is around 3,27,000. And in the later stage of growth, the result is an explosive increase in cell numbers. Because of that, the gap between 2 points is close here and the gap increases in this area. And if the culture incubated more, you can't feed the graph on a single page. Not only that, suppose you want to know the bacterial number at any point from this graph, you have to use this equation. And the calculation is more complicated. Lastly, the bacteria are growing exponentially in this region as well as in this region also at the same speed. But from this graph, we can differentiate that it looks like at the beginning they are growing slowly and here they are growing very fast, but that is not the actual case. In order to solve this problem, the number of cells is plotted on a logarithmic scale and time is plotted arithmetically, and that's why this graph is called semi-logarithmic plot. The point falls on a straight line. The straight line function reflects the fact that the cells are growing exponentially and the population is doubling in a constant time interval, and here in this area also. Semi-logarithmic graphs are also convenient to use to estimate the generation time of a microbial culture from a set of growth data. The equation of the straight line is also very simple. Now let's check how we can measure the value of G and K from this plot. From this graph, we can calculate the value of G in two ways. Generation time may be read directly from the graph. For example, when 2 points on the curve that represent one cell doubling on the y-axis are selected and two vertical lines drawn from them to intersect x-axis, the time interval measured on x-axis is the generation time. You may ask why. Here is your answer. We know that the time required to double is called the generation time, or G is T by N. And here in this graph, N, or the number of generation, is only 1 from 20 to 40. So here N is 1. Or we can say that G is equal to T when N is 1. For organism A, from 20 to 40, it takes 1 hour. So for organism A, the generation time is 1 hour or 60 minutes. For organism B, from 20 to 40, it takes 30 minutes. So the generation time is 30 minutes. In this way, you can calculate G directly from this graph. Now the second way, here g is 0.301 divided by slope of the straight line. If the slope of the straight line is provided, you can easily calculate g. Let's check. For organism A, the slope is 0.301 and for organism B, the slope is 0.602. So for A, g is 1 hour or 60 minutes and for organism B, g is 0.5 or 30 minutes. Here is the third method. From Excel, how you can determine g. First, I have plotted this data. Double click in this region, then format axis window will open and change the scale to log base 10. Then go to add chart elements and then trend lines and click onto more trend line options. Click exponential and then click display the equation on the chart and display r square value on the chart. You know that the equation of the exponential line is y is a into e to the power bx. Here b is 0.6931, so natural logarithm 2 by 0.6931 or 1 hour or 60 minutes. Here the values are in hours, so our result comes in hour also. We will convert it into minute. Do you know, you can also differentiate which organism is growing faster from this graph. If the slope is large, the organism is growing faster. Here organism B is growing faster than organism A. Generation time short means the slope of the straight line will be large. So that's all for now. In the next video, I will describe batch growth characteristics and batch growth curve. If you like this content, subscribe to my channel. You can share the link with your friends, and finally, thanks for watching.